Hello, everybody. If you're attending in person or on Zoom or watching this in the future even, welcome to the first Careers in Genomics Symposium hosted by community organizers in genome sciences. Some people from COGS over here. So my name is Steven Gonzalez. I'm a second-year PhD student in the Trapnell Lab. I'm studying vertebrate embryogenesis. I'm also the leader of outreach and community organizers in genome sciences, which is a student-led organization in the department that fosters engagement to make people feel welcome in genome sciences, regardless of their background, and engage with the community of UW and beyond. And so, here we go. So first off, I'll be giving a quick intro to the event goals, a little bit about my personal background, why I thought that this event would be worth holding, and then a very short intro to genomics, and then a little personal story about genetic variation in my family and heart disease. So first off, what does this event aim to accomplish? So what I really want you guys to take out of this is, or take away from this is, to clarify your research interests, to know kind of what's being done, who's doing it, why it's significant, and then also to learn about some paid opportunities in research, such as the NSF research experience for undergraduates, LSAMP, Louis Stokes Alliances for Minority Participation, and the GRTDS program. And so the way that I wanted to accomplish this is that for clarifying your research interests, I wanted to have some short accessible talks that are presenting really cool research done by my friends in genome sciences. There's going to be an info session where you guys will be able to learn about these 3 programs. And then there's also going to be a career panel where we have we've got 4 people from different backgrounds that are going to be talking about their experiences in both research and academia and industry. So first off, personal background. I am from Southern California, from a little beach town called Encinitas. So Encinitas is known for really good surfing and fantastic Mexican food. I've never gone surfing in my entire life, but I've definitely eaten a lot of really good Mexican food. And so when I was in San Diego, I went to Miracosta Community College for a few years. And when I was there, I got involved in the honors program at Miracosta. But during this time, I found it really challenging to actually get a job as like a lab technician in industry or academia. And so I was working at Legoland for a little bit, and then I also worked at a used car dealership. And so when I transferred from Miracosta Community College to University of California, San Diego, I started working at the Salk Institute in the lab of Joe Ecker, studying plant biology, how plants respond to drought stress. Eventually, after 3 years of working as a technician, I decided that I wanted to pursue a PhD, and I decided to pursue that PhD at Genome Sciences at University of Washington. But, for the first time, I was kind of faced with this question, like, what should I research? Like, you have to select a lab, and you have to have like a general idea of what everybody's doing to make an informed decision of which lab you're going to join. And then once you join a lab, then you might have to work on like you're going to work on a unique project. And so it can kind of take some time to figure all that stuff out. And so ultimately, I wanted to kind of have an event where I could, have you guys get some exposure to a lot of different research. And ultimately, I decided on Cole Trampnell's lab, where I study vertebrate embryogenesis. And so here we have, this is actually a zebrafish that's been injected with mRNA encoding stay gold green fluorescent protein. And so it's injected at the single cell stage. So these are all the cells, and they're going to continue proliferating and moving, enveloping the yolk. And then we end up with this, so the glowing zebrafish. And so on to the intro to genomics. Within every single one of your cells, you have half of your genetic material is from your father and half of it is from your mother. And ultimately, this is going to control a lot of different traits, such as your height, eye color, your diseases you may be susceptible to, and a lot more. And so The cells that are comprising your body contain the entirety of your DNA. So there's 3 billion different base pairs. There's 21,000 genes that encode instructions for building proteins with unique functions. This is only 1% of your genome. A whole lot of other sequences encoding the logic of where and when these genes are expressed. So that's 99% of the genome. We'll be talking a lot about different coding and non-coding variants today. And so ultimately, we have a lot of different cell types that are in our bodies that are playing diverse functional roles. And so these different cells are expressing different genes. So like, for example, myocytes are involved in contracting and allowing us to move our bodies. Pneumocytes are facilitating gas exchange. Chondrocytes are making cartilage so that we can have, frictionless movement of our joints. Neurons are transmitting information. Erythrocytes are holding oxygen, and osteoblasts are giving rise to bone. And so these are all expressing very different genes. And ultimately, it means that different variation in these genes could affect very specific tissues. And so what is genomics? So it's the study of all genes. So this suffix of the omics or genome basically implies totality. So we're interested in studying all genes. And so I'm just going to put up some questions of what people are interested in studying in genomics. So how does variation in our genes affect our health? How does variation in our genes affect our response to medication? How does a diseased cell differ from a healthy one? And if we can characterize a disease state, then we can diagnose people and potentially develop therapeutics that are going to reverse a diseased cell to a healthy one. How do cells utilize a subset of genes in the genome to carry out all their functions? And do different organisms use the same genes for the same functions? Could variation in these genes affect very specific tissues? And another big question is what genes are required where in development? How could variation in these genes cause developmental disorders? So that's a very, very brief intro to genomics. People are going to talk more in detail about different aspects of these questions in the talks that will come after mine. So now I'll talk a little bit about a personal story about genetic variation in heart disease in my family. And so bicuspid heart valves are extremely common in my family. In this picture, there's actually 3 people with bicuspid aortic valves. My grandfather, my mother, and my brother. And so you can see this image on the right. A normal aortic valve has this tri-leaflet design. And so this ensures that blood can only travel one direction. If you have a bicuspid aortic valve where it's gone from a tri-leaflet to a bileaflet, then you can have regurgitation in which the blood that's pumped immediately out of the heart is going to go backward, and that's not really efficient. And so it's very, and so like here we can see 2 pictures of like some other family members that have bicuspid heart valves. And so clearly there's some genetic component to this heart disease because it's so prevalent in my family. And so bicuspid heart valves can actually be caused by defective notch signaling. And so there's this Notch1 receptor that is expressed within the normal aortic valve. And so notch signaling through this Notch1 receptor is going to be prohibiting the expression of RUNX2B. And so RUNX2B is a gene that is expressed during ossification, or when things are mineralizing and turning into bone. And so ultimately, if you have some sort of variant in Notch1 in this receptor, and you are perturbing notch signaling, then we get the expression of RUNX2B, and it leads to calcification of the aortic valve, and so it becomes a bicuspid valve. And so in late 2023, my brother actually had an aortic aneurysm, which is something that can occur when you have a bicuspid heart valve. And so this was a pretty awful experience. So Notch1, this protein contains 2,555 amino acids, so it's pretty massive. My brother's variant is a proline to serine nonsense mutation from at this 832nd amino acid. And so ultimately, these are both neutrally charged groups, but proline is hydrophobic and serine is hydrophilic. And so what ends up happening is that it's very possible that this specific variant could be changing the protein structure a ton, and we're no longer getting Notch signaling where we should have it. And so lo and behold, mutations in Notch1 are known to cause aortic valve disease. And furthermore, some Notch1 mutations are associated with aortic valve disease and aortic aneurysms. And so we actually found that there's one other patient in this online resource called ClinVar that has this specific P832S mutation, and they also have had aortic aneurysms. And so this variant, though, is classified as there's an uncertain significance. So we're not sure if this variant is actually causing aortic aneurysm or bicuspid heart valves at all, but with some further functional characterization, then we could be able to prove that this variant is specifically causing this disease. And so this is only we're only talking about one gene here, the Notch1 receptor. And so with 21,000 different genes that can all interact with each other, then there's a lot of potential for our genetic variation at scale to affect our health. And so our first session is going to feature speakers that are studying the effects of genetic variation: Lizzie, Malvika, and Hank.